Hi, my name is Madison Eastridge, and today I'm going to be talking about Chapter 8, Microbial Genetics. So within Chapter 8, Microbial Genetics, there's three different processes that we've been learning about in class. First one being DNA synthesis, next is transcription, which is RNA synthesis, and then the last one is translation, which is protein synthesis. So starting with DNA synthesis, when a cell replicates, it needs to double its DNA so that it has two things of DNA to go into each cell. So when the DNA replicates, it's going to have a parental strand and a daughter strand. The parental strand is the strand that it originally had, and then the daughter strand is the new strand that it is forming. So DNA is replicated using DNA polymerases, and those are just enzymes that help the synthesis of DNA. So starting is DNA gyrase, and DNA gyrase's job is to uncoil the DNA. So after the DNA is uncoiled comes DNA helicase, and DNA helicase's job is to unzip the DNA, and that's why it's going into separate directions right here, and this right here is called the replication fork. So up here is the leading strand, and down here is the lagging strand. This is called the leading strand because when DNA replicates, the enzymes need to go in a 5' to 3' manner, which is what is happening up here, but down here it's going from a 3 to 5, so it's kind of harder, so it can't go continuous, so it's going to have little nicks in there, and that's when this DNA primase comes in. So DNA primase is going to put RNA primers, just for time being, in the little nick slots, and then after it's going to come DNA polymerase 1, and its job is to fill the RNA primers into DNA fragments called Okazaki fragments. And those are not going to connect all the way with the DNA ahead of it until DNA ligase comes along. And that's DNA ligase's job is to join the two things back together. So up here is always DNA polymerase 3, and it's just going in the 5 to 3 strand direction, replicating the strand of DNA. Moving on to transcription. Transcription is RNA synthesis, so it's making RNA. It can make mRNA, rRNA, and tRNA. So mRNA, for example, might go, it's like messenger RNA, so it might carry like coded information from like the DNA to the ribosomes. rRNA is going to go to ribosomes, and then tRNA could transfer like amino acids. And so what this is showing right here is this is the DNA, because DNA has to be a template for RNA. So what it is like they're similar to each other with the nucleotides, but RNA nucleotides don't have thymine. They use U instead, and so right here you can see like A represents pink, T is blue, C is green, and G is purple. So those are the nucleotides of DNA, but then when RNA polymerase comes in, it's going to change those T's to U's, because U is an RNA nucleotide. So DNA polymerase comes in at a promoter, and that is attracted to the DNA strand. And so this RNA polymerase is going to continue down the DNA strand using it as a template to make the new RNA until it comes to a terminator gene, and that is going to tell it to stop replicating, and that will be the end of the RNA strand. So then that's how you get your RNA strands. And so finally is translation, and translation is protein synthesis. So what it is, it's just the making of proteins. You're using amino acids and bonding them together with peptide bonds in order to make protein chains. So these two black shapes here are represented the ribosomal units, and it comes together with mRNA and tRNA. And what it's doing is, so the first tRNA will come in, and it'll go in the A slot, and then the P, and then the E, and then it just keeps going down and pushing this way. So the unit will go this way, and the mRNA just stays. And so when the mRNA comes in, it's already going to have amino acids attached to it. So this is the start codon, and the start codon always starts with AUG. And so there's going to be like other codes within the mRNA fragment. And so say this was just a single fragment itself, the end codon here is UGA. And there's three things, end codons you can have. For example, it can be UAA, it can be UGA, or UAG. But those are the only options for end codons. And again, AUG is the only option for start codons. So when the ribosomal unit comes to those different codons, it knows like what to do rather than to start or to stop. So then when these amino acids come together, these ribosomal units, like I said earlier, they are connected with peptide bonds to form together to make a protein. So after it's done and the protein is made and it comes to an end codon, the parts will then come apart and just to go back to their different ways. So I've reviewed DNA synthesis, transcription, which is RNA synthesis, and then translation, which is protein synthesis, all three processes that are part of Chapter 8 Microbial Genetics.